Hi everyone, my name is Karin M Saltoun and I'm a graduate student here in Dr. Dinanob's Lab at McGill University, affiliated with Mila AI Institute in Quebec. Today, I'm going to talk to you guys about brain asymmetry. So, as a bit of background, a lot of people think that the two sides of the brain are either completely identical or uniquely subserve different functions. The right side of the brain is often associated with artistic and creative thinking, whereas the left side of the brain is more associated with logical and analytical thinking. As a fun fact, these ideas are so steeped in our society that they even make it to the dictionary definition of left and right brain, respectively. Yet, despite how prevalent these ideas are, is such a simple viewpoint really the best way to view asymmetry? Over the decades, brain asymmetry has been linked to both spoken and written language, handedness, sex, visuospatial attention, and more. Yet, despite how relatively well-established functional asymmetries are, even as recently as 2018, there has emerged little consensus as to which anatomical asymmetries even exist at the population level. Digging into the literature, you'll find that researchers often focus on the cortical shell, identifying pairs of homologous brain regions, i.e. regions that are the same on both hemispheres, and asking are they larger on the left or the right and to what degree. Implicit in this is the idea that there's a single archetypical axis of asymmetry. Here, I use axis of asymmetry not to refer to a spatial axis of left-right enlargement, but to a collection of regions who have consistent asymmetry across a population. In this viewpoint, all people and all functions lie along this one archetypical axis of asymmetry, with everything else being attributable to inter-individual noise. Today, I'm going to put these ideas on a back burner and take a new perspective on brain asymmetry, namely investigating the possible existence of multiple distinct axes of an asymmetry. In this view, there are several archetypical asymmetry axes which coexist across a population. A natural advantage of this is that we can ask the traditional question on each of these axes separately, and this may naturally segregate different functions to different asymmetry axes. In order to investigate this question, I drew upon the UK Biobank resource, a repository of 40,000 middle-aged individuals recruited across the UK, involving uniform MRI acquisition, processing, and registration to symmetric atlases. This allowed us to take homologous brain pairs and compute a unitless scaled measure of both degree and direction of asymmetry. In doing so, I went from 170 atlas parcels to 85 targeted measures of brain asymmetry, spanning cortical, subcortical, cerebellum, and white matter tracts. Armed with this information, I applied a dedicated machine learning pipeline to identify common whole brain patterns of asymmetry, which pervade and persist across the whole brain. With a single model, I was able to identify one or many common motifs of asymmetry which emerge in the population because our machine learning pipeline associated an importance score to each of our discovered symmetry patterns. This importance score looks like this, and to orient yourself, each point in this graph represents a single asymmetry pattern, with position on the y-axis indicating how much of the underlying asymmetry in the population is captured by this one asymmetry pattern. In all, we find 33 separate asymmetry patterns are able to explain symmetry structured above and beyond what can be captured by a single regional asymmetry alone. By construction, each of these asymmetry patterns are uncorrelated from one another, meaning that individual expression levels of pattern 1 is unrelated to individual expression levels of 2, 3, 4, etc. This thus supports our idea that there are multiple unique axes of asymmetry which exist across the population, but how can we test if these patterns are whole brain? In order to do that, let's zoom in on asymmetry pattern 1. Here, we are looking at the co-occurrence structure of pattern 1. All of these different rows represent different levels of brain asymmetry. Captured by asymmetry pattern 1, but again, this is one pattern which came of the many that emerged within our model. The darkness of brain features indicate how important they are in describing asymmetry pattern 1, and from this we see there's a strong involvement of brain features across all examined levels of brain organization. Brain features which tend to show shifts to the same hemisphere are plotted on the same hemisphere, and to demonstrate this idea, I'm going to use the frontal pole as an example. We see that asymmetries in the frontal pole co-occur with ipsilateral shifts in the temporal pole, but contralateral shifts in the occipital pole. We're looking here at relative and not absolute asymmetries. That is, it's equally valid to read this as left frontal, right occipital enlargement, or the reversed pattern of right frontal, left occipital enlargement, with the important thing being that these brain regions are contralateral or ipsilateral, depending on if they're plotted on the same or opposite hemispheres. In particular, the pattern of contralateral frontal and occipital enlargement sounds an awful lot like brain torque, which is one of the most well-documented structural asymmetries, captured by the idea that the one hemisphere of the human brain tends to be shifted slightly posterior to the other, leading to the protrusion and enlargement of the right frontal and left occipital poles. This phenomenon is well documented in humans, but not in chimpanzees, and thus may be human-specific. Important for us, our machine learning model knew nothing about this particular structural configuration, but was able to naturally extract contralateral frontal and occipital pole enlargement and connect it to asymmetries in the cerebellum, white matter tracts, and temporal pole. Beyond that, I also capitalize on the fact that the biobank involved a number of lifestyle, health, and cognitive testing, which allowed me to extract nearly 1,000 behavioral and lifestyle traits spanning 11 broad domains. I then associated the individual asymmetry pattern 1 expression to each of these phenotypes in a phenome-wide association study, and the negative log strength of associations between asymmetry pattern 1 and all of these phenotypes is shown here. And we see that asymmetry pattern 1 is strongly associated to sex-related hormones, hair balding, family size, and in that physical general gray category in the middle, a lot of anthropometric features. I've also taken the liberty to highlight right and left hand dominance in those red and blue crosses in the gray physical general category, showing us that asymmetry pattern 1 is strongly associated with hand dominance. However, we see a notable absence of cognitive and mental health phenotypes, despite knowing that brain asymmetry may potentially be related to language. But as a reminder, though the literature is dominated by investigations to brain torque, we found several other asymmetry patterns which explain quite a bit of structural asymmetry variation within the population, but have thus far been unexplored or even unable to be found in other research. So let's zoom in on asymmetry pattern 2. Here we see the co-occurrent structure, and again, we find that there's a strong involvement of brain features across all different levels of investigated brain organization, including contralateral, planum temporale, and cerebral lobe 6 asymmetries. Some of you may know planum temporale as Wernicke's area, which is one of the most prominent structural asymmetries found in human brains. Here we find that planum temporale asymmetry does not occur in isolation, but rather is linked to cerebral lobe asymmetries. In our phenome-wide profiling, we find that individual asymmetry pattern 2 expression is associated with many mental health and cognitive phenotypes, including language-related questionnaires. We also see associations with several socioeconomic status, markers, blood assays, but not right and left hand dominance in that physical general category. In the interest of time, I've summarized our top 4 asymmetry patterns here, and including brain features that are prominently highlighted in each. We see that the planum temporale is involved in multiple distinct asymmetry, indicating that asymmetry in the planum temporale is separately associated with both cerebral and hippocampal asymmetries. We also find that each of our asymmetry patterns has a unique phenotypic profile. Overall, we find that brain asymmetry is well described by multiple whole brain patterns of asymmetries, which integrate regional asymmetries from across the brain. Each of our discovered asymmetry pattern had a unique phenotypic profile and was able to recreate known principles of brain asymmetry. Thus, our discovered asymmetry patterns can potentially serve as a scaffold for future investigations into brain asymmetry. I'd like to thank my friends and colleagues here at McGill University, and I look forward to hearing any of your questions and answering any of your questions. Thank you very much and have a great day.